Welcome back to the lecture 4 of our cell series. In this video, we will explore the cell wall and the endomembrane system in detail. So let's firstly start with the cell wall. Now the cells of the bacteria, fungi, algae and plants have a rigid coat, the cell wall, which is present outside the plasma membrane. The composition of cell wall varies in different groups. In bacteria, the cell wall is made up of peptidoglycan. In plant, the cell wall is the outermost layer, it is the non-living layer. In algae, the cell wall is made up of cellulose, galactans, mannans, and minerals like calcium carbonate. In plants, cell walls consist of cellulose, hemicellulose, pectin, and proteins. So let's study about the structure of the cell wall. The cell wall consists of two regions, primary wall and secondary wall. So the first layer formed by the young plant cell is known as the primary cell wall. It is thin, elastic, and capable of growth, which gradually diminished as the cell matures. Now, in mature cells, more layers of wall material added to the internal of the primary wall. These layers are called as secondary cell wall. Now, secondary wall have been derived from the primary wall. Now, secondary wall is rigid and incapable of expansion. Now, middle lamina is a layer which mainly consists of calcium pectate, which holds or glue the different neighboring cells together, the cell wall. So, the dark portion here is the primary cell wall. And then there is plasma membrane present and secondary wall present inside here. This is about the structure of the cell wall. Next, we have the functions of the cell wall. The cell wall helps in maintaining the shape of the cell. It protects cell from any mechanical injury. It wards off the attack of any pathogen. It allows material to pass in and out of the cell. There are breaks in the primary wall of adjacent cell through which one cytoplasm of one cell remained connected with the other. The cytoplasmic strand which connects one cell to another is known as plasmodesmata. Now see in this diagram, two adjacent cells are connected through the cytoplasm with the help of plasmodesmata. This is all about the cell wall. Next, we have the endomembrane system. So, the endomembrane system consists of endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. Endoplasmic reticulum is a singular membrane-bound organelle. It is a network or a reticulum of tiny tubule structures scattered in the cytoplasm. Now, the intercellular space divides into two distinct compartments: luminal, which is present inside the endoplasmic reticulum, and extraluminal, which is present outside the endoplasmic reticulum, which is cytoplasm compartments. So there are generally two types of ER. First is rough endoplasmic reticulum, also known as RER. Then there is smooth endoplasmic reticulum, which is known as SCR. Its surface is rough due to presence of ribosomes. The function of RER, it involves in the process of protein synthesis and secretion. Second, we have SCR or smooth endoplasmic reticulum. Its surface is smooth due to the absence of the ribosomes. Function of smooth endoplasmic reticulum, it is the major site of lipid synthesis in animal cells. The lipids are like steroid hormones are synthesized here. Now we have endoplasmic reticulums diagram. Now endoplasmic reticulums remain in contact with the nucleus. So let's see the diagram. Rough endoplasmic reticulum, granules-like structures which are known as ribosome, which helps in protein synthesis. And SCR do not contains ribosome. These are called as agranular structures. And there are cisternae present and cisternal space. This is all about endoplasmic reticulums diagram. So let's start with the Golgi apparatus. Now Golgi apparatus has been first observed by Camillo Golgi. He observed a densely stained structures which are later named as Golgi bodies. They consist of many flat disc-shaped sacs or cisternae. These are stacked parallel to each other. Cisternae are concentrically arranged with convex cis-forming face or concave transforming or mature facing. Both faces are interconnected. Now the functions of Golgi apparatus are it performs the function of packaging materials, protein synthesis in the ribosomes in RER are modified in the cis phase of Golgi bodies before releasing into the trans phase. Now next we have is lysosome. These are membrane-bound vascular structures formed by the process of packaging in the Golgi apparatus. It is found in the almost all plant cell except RBCs of mammals. It is very rich in all types of hydrolytic enzymes like hydrolase, lipase, carbohydrates, and protease, and optimally active at acidic pH. These enzymes are capable of digesting carbohydrates, proteins, lipids, and nucleic acid. Now, in the diagram, a vesicle has been released from ER, which has been modified in the Golgi apparatus, and it is known as the primary lysosome. Primary lysosome may be a pinacocyte vesicle or a food vacuole or an autophagic vesicle. This can be entered through the cell wall and it's modified into the secondary lysosome. This is about the lysosomes. At last we have vacuole. Now vacuole are the membrane bound space found in the cytoplasm. It contains water, sap, excretory products and other material which are not useful for the cell. It is formed by the single membrane known as tonoplast. Now in plant, it can occupy 90% volume of the cell. In plants, tonoplasts facilitate the transport of number of ions and other material against the concentration gradient into the vacuoles. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In protists, food vacuoles are formed by engulfing the food particles. This is about the today's lecture. I hope you like it. Thank you for watching.